I want to thank everyone for joining us here tonight. It's a tremendous pleasure for me to be able to introduce Professor Donald Pfister. And when thinking about what to say tonight, I decided that I wanted to start with a confession. And that confession is that last night, for the first time, I nearly joined the world of social media. So that's right. I actually I almost started a Facebook account. And now, for a variety of reasons, I've resisted this temptation for years, actually. But before I tell you why I nearly joined social media, I wanted to give you a little background. So many of you will know that Professor Pfister wears many hats, and he has, over the course of the 40 years that he's been at Harvard, worn a number of them over this period. And in addition to his role as professor, he is also the curator of the Farlow Library and Herbarium of Cryptogamic Botany. And for, I think, nearly 30 years on and off, he was the director of the Harvard University Herbaria, of which the Farlow is one important collection. He was also the former co-master of Kirkland House, along with his wife, Kathy, who's here this evening. I saw her somewhere. Yes, right there. Thank you. He was co-master for 17 years, which, as many of you will appreciate, is a labor of love. Now, if this isn't enough, last year Don was appointed the interim dean of Harvard College, and this is really the part of the introduction where I want to return to my story about social media, because it was only yesterday that I found out that Don has a Facebook page. Now, this isn't just any sort of Facebook page. This was lovingly created and curated by undergraduate students. And these undergraduate students are affectionately known as Pfister fans. Fans is spelled P-F-A-N-S. So this is an homage to Don, of course. And he has nearly 2,000 likes on this Facebook page. But all this is to say that during his time as dean, Don really became a rock star in this role. And it was really in the context of the Facebook page, which it turns out I was able to read without joining Facebook, because they were mostly publicly, most of it. It was mostly publicly available. But you can see in the contents and the sort of impressions of the lives that he touched in so many different ways. So not only was he delivering cupcakes to students during exam time, but he wrote regular emails to students, and they included a variety of things, including reflections on his childhood growing up in the Midwest, witty remarks about recent events around campus that he had attended, and he even inspired actually a student-led book club, which was sort of featured the books that he was reading at the time. And yes, he even wielded his power to unleash his love of fungi on these young and impressionable minds at Harvard College. So here he pointed out the magic of fungi, and indeed in some cases of plants that are all around us, including on the Harvard campus. And I think what this importantly did was to sort of bring a sense of lightness and of joy when it was needed most. So it was, of course, during the darker times that he sat with students during a bomb scare and consoled them when one of their classmates had died. So, and the students really loved him for this, and it all really actually came to sort of a head, I think, at commencement. And I was fortunate to have been on the stage at that time. And when Don rose to confer the undergraduate degrees to these students, it was a roar, right? And that roar was followed by a chant. And that chant was, "Pfister, Pfister, Pfister." I mean, it was an impressive and emotional moment, and I think even Don was overwhelmed and touched by this, and I felt very proud to have been there. And I think that the reason why the students love him so much is just a testament to how he's viewed by so many of us. He's wise, he's steady, he's patient, and he's long been a quiet sense of and source of inspiration for us all. And I personally am tremendously thankful to have known him for nearly 20 years since I started as a graduate student here. And over the last 8 years, I've been privileged to teach with him. We co-teach a course called Plants and Human Affairs, which happens to be one of the oldest running courses on campus, so I'm told. And he also teaches, though, the biology of fungi, the biology of trees and forests, and a mycology course for graduate students. And if that's not enough, Don has taught in the summer school, where he was also dean for a period, as well as the extension school, where he's taught for more than 25 years. And so tonight, you've come to hear stories about Don's research. And his interests are broad. As you'll see, his research centers on the biology and systematics of fungi, and he uses molecular approaches, life history information, and morphological methods to understand fungal relationships, to discern their biogeography, and to elucidate their hidden and fascinating diversity. He's also has an active interest in collections and collectors more generally. Don has been the recipient of numerous prestigious grants for his work. He has served as both president and fellow of the Mycological Society of America. He received the distinguished William Weston Prize for Excellence in Teaching. He's a AAAS fellow, and he has mentored a distinguished group of national and international students. So I could go on, but I won't. And instead, I'll leave the stage to Don. Thank you, Don.
Well, now that Chuck has set the stage, I get to unset it a little bit. I'm not talking about my research tonight. Sorry. I'm talking about collections and how we can use those and ways in which we think about the collections as a part of our heritage, a way in which we can use these to reflect upon the importance of collections in our, if not daily life, in our collective life. So this has this daunting title under here about where I've spent the last 40 years, which is the Farlow Reference Library and Herbarium of Cryptogamic Botany. And I want to take a moment to kind of dissect this, to give you a sense of what this all means, because some of it is apparent and not all of it is. But since we're celebrating this institution, I want to make sure that you have the pieces of the puzzle together that come from this title. So first of all, Farlow. Farlow is a man, William Gilson Farlow. And I give you his dates here. He died in 1919, and it's because of his bequest that we have the institution that bears his name. And I inserted this little piece at the bottom here that is a snippet from a letter that I discovered when I was at the Lloyd Library in Cincinnati, Ohio. And he sent the letter along with his photograph here, a photograph from 1916. And he says, I'm sending with this my photograph, which some people say is good, but some do not. At any rate, I'm always satisfied, because not to be satisfied would mean that I must sit again, which I should never do if I could help myself. In the photograph, I seem to have but one hand, but that one, you will see, is arranged in the most approved manner, so that I appear to be lost in thought, perhaps thinking, what an infernal bore it is to have one's picture taken. So this is Farlow. He was a protege of Asa Gray, studied in Europe with Anton de Bary, came back to the US, and I'll mention a little bit about some of his research and some of the important things that he did in a while here. But his contribution was that he was the first professor of cryptogamic botany, and I'm going to define that for you in a second. in the New World. He studied fungi, he studied algae, he studied lichens a little bit. He didn't like mosses much, but those are the groups that we're going to be talking about. So reference library, what does that mean? What does it refer to? I mentioned that when Farlow died, he left Harvard a bequest. The bequest was his personal library, as well as funds to support activities in this field. And his personal library consisted of many, many volumes, and many of them were unique in North America. That is, as a collector, he found things that were very, very special, and he collected them in his private library. And the library today, and the building today, the roof today that we're talking about is at 20 Divinity is the real address. You enter via 22. It's a small Victorian building that you see from the Biolabs courtyard. And if you take in this, it's quite an impressive building. It was built as the library for the Divinity School and was converted into the collection space as we know it now in the 1920s. And it's this opening of this building that we're celebrating today. And again, a beautiful building and full of treasures, full of all sorts of interesting, unique, and special materials, books, journals, and an archive. And you'll see some examples from all of these tonight. Herbarium. Herbarium's a little bit of a misnomer for the collection that we have here because they're not herbs, of course. But the herbarium is the collection of specimens, and here you see a cabinet. All of these yellow sheets indicate that there are specimens out on loan. So part of what we do is store and have these important materials, the actual specimens. But these then are used by scientists from around the world, and you can see in this particular section here that it's highly, highly used, many, many specimens out on loan. And then I'll point out the specimen here, and you get an example of Farlow's own handwriting here. Not so neat, but it's Peronospora viticola. And I pulled this out. It's collected at the Botanical Garden in Cambridge up on Garden Street in 1874. And it's at that point that Farlow was beginning to work back in the US after his time in Germany. And he was at the Bussey Institution. The Bussey Institution was kind of as close as Harvard got to an ag school. And so he began studying plant pathogens. One of the pathogens he began to study was this one, which is a grape pathogen, a downy mildew. We get to those M's, the downy mildew of grape. And he did quite an extensive study of the downy mildew of grape. It's a parasite. This fungus-like organism is a parasite on the Concord grape. And he makes a wonderful comment there. As the ripening, as the sun shines upon the plants with its ripening rays, the leaves wither because of the infection of this organism, and the grapes are exposed to these ripening rays of the sun. Should it be introduced into Europe, it might be a problem on the European grape. And so within about 10 years, it was introduced into Europe. The grapes were nearly lost in Europe, but Farlow's premonition here of what might happen was an important piece of thinking about the biology of this organism. So here's one of the specimens from the beginning in that study. And then this tongue twister of cryptogamic botany. What is that? Cryptogamic botany. Well, cryptogams refers to these organisms that reproduce by spores. They don't have flowers, they don't have seeds, and we think of them as the ferns, which have vascular systems. We're not talking about those, but mosses, fungi, algae fall into that group. So hidden union, hidden reproductive mechanisms. And these are not to be confused. If you're typing and your computer is telling you how to spell that word cryptogam, it will tell you it's cryptogram. That's wrong. And I want to make sure that you remember this forever. That's a cryptogram. It's a very simple shift of letters. So B becomes A, and it shifts through the alphabet. None of you who've been watching me do this on the blackboard should answer. Who's quick? Who can tell me what this says? All right, you give up. What would a mushroom car say? Okay, a cryptogram, not one of these organisms, but a cryptogram. Okay, so the Farlow Reference Library and Herbarium of Cryptogamic Botany. And the doors opened at the Farlow November 28th of 1924, and this is the guest register from that first day. And you'll see the first signature there is Mrs. W.G. Farlow. And we've been reflecting on this a little bit in the last couple days. And in the archives, there's a wonderful letter from Mrs. Farlow to Roland Thaxter, who was instrumental in making this all happen, saying she spent the afternoon there and she felt that William Farlow was there with her. The others on that day are important botanists, some studying lichens, some studying fungi, but I will also point out that a few days later we had a very important visitor here, Franklin Roosevelt. So a place where botanists have come and mycologists and others for generations. So now, just what are those kingdoms that I'm talking about? I say one roof, and we have the roof at 20 Divinity Avenue. You've seen a little bit about the collections in the library. But what about the organisms that are there? And I like to think that the Farlow is a really special kind of collection because we have so much diversity, so many kingdoms that are represented within the collection. And here's one way to look at this. You see the plants out there on the side. They're a really pretty insignificant part of the diversity of the world. Animals are here. They do all right, but they're not that diverse either, except for the beetles. And then the fungi, and the fungi are really, really diverse. We think that there is somewhere in excess of 2 million, perhaps, fungi. We've described only about 5% of those fungi, so there's a lot to describe there. And then there's these groups, the protista, that represent a number of simple in terms of organization, multicellular organisms often, without high organization, and then the bacteria and the archaea. So I want to kind of work on this theme and talk about some of the organisms and tell some stories about some of these organisms that are housed within the Farlow collection. But first, this may be a way that we're taught or think about the organization of life. This is a very simple chart, and it's not a tree of, it's not an evolutionary tree, but rather early on, Edward Hitchcock, who was president at Amherst College, was a geologist. And he went about saying, so when did certain groups arise? And so this is really telling us when groups arose. But I think it's so wonderful because you see, the kind of pinnacle with the plants. You may not be able to see that, but those are the palms. So we're all aspiring to something here. And the pinnacle for animals, of course, is man. But you'll see that these are unconnected units here, the plants and the animals. And along came Darwin. This is the famous sketch from his sketchbook. And what Darwin is pointing out here is that all life is derived from a common ancestor. So down at number 1 is the common ancestor, and then there's diversification over time to give us the great tree of life that we see today. And we can see that in another representation. Many of you may have seen this. This is Haeckel's chart, and showing us that here's the tree, but realize that all of those branches originate down here from a common ancestor. So it's the monophyly of life. And you can't read that, but let me outline the groups that I'm going to talk about and that I will include tonight. And so they are here, and you see that they represent some of these branches in Haeckel's tree. I'm a little disappointed always because when I look at this, because over here looks like it's a very insignificant part of the tree. These are fungi and these are lichens, and that doesn't sit well with me. Here's another way of representing this tree, and I'm going to use this as a guide as I talk about various groups and various examples here. And again, here are all the branches, and here's the common ancestor. And so a number of lineages, a number of units that we can look at here. And these red splotches that I've put on here will be kind of guideposts for what I'm going to talk about, where these organisms that we collectively call cryptogams hang out. So what about those mummies? I want to talk a little bit about real mummies, an Egyptian mummy, and how that might connect here. And then there's some usage of the term mummy that we can think of as more figurative, perhaps. And of course, when we talk about, what I'm talking about first here are lichens, a lichen example. And lichens are fungi teamed up with green, in this case, with a green alga. So this is where we are on our phylogenetic map. And we're thinking about a symbiotic relationship here. Now, my colleague, Michaela Schmuel, and who's here, followed up on this story that we have of our oldest collection. So this is a lichen. Here's the actual specimen. And it was unwrapped from a mummy, where it was used as part of the processing, the mummification. And it was unwrapped in 1859 in Canada. Apparently, she discovered, mummy unwrappings were kind of a party event. You buy your mummy and bring it back, and then you have a party and you unwrap and see what you have. And that's what they were doing in 1859. And Mr. Poe took some of the lichen and sent it to American lichenologist Edward Tuckerman. And I always pause with Edward Tuckerman. Edward Tuckerman, you know Tuckerman's Ravine up in the White Mountains? Edward Tuckerman is that guy. And he was an explorer in the White Mountains. He studied lichens. He was the kind of premier lichenologist for the middle part of the 19th century in North America. So, of course, the material that came from the mummy, and if you wanted to work this out, would go to this famous lichenologist, Edward Tuckerman. And it did, and you can see what they say about it here. And this is from the Tuckerman Herbarium. His name was not Herb, it's abbreviated Herbarium Tuckerman. And his scrawly little handwriting. And I'll point out that as you look at these specimens, realize that we as curators become really good at guesswork on handwriting and locations. That we really have to become excellent with this. So the lichen turns up in Canada, it's unwrapped, and the lichen ends up with Tuckerman. And the question that Michaela asked was, so does that lichen grow in Egypt where the lichen turned up, or where the mummy turned up? And the answer is no, it doesn't grow there. And if one begins to expand here, what's outlined in red is the possible origins of the lichen. So the lichen grew outside of Egypt but was brought in, as were other commodities that were used in the mummification process. Pepper from India and frankincense and myrrh with the purple lines. So not only do we have a kind of use for lichens here that is ancient, but it also sheds light on these trade routes and what must have been going on at that time. So it's a really interesting story, this lichen, old lichen that we have in the mummy. Now, mummies are not restricted to lichens or to mummification of Egyptians, but there are some fungus mummies too. And some of you may recognize some of this. This is you may know it as cordyceps sinensis. Ophiocordyceps is the name that's used these days. This is an insect parasite, and if you look really closely, you can see the insect part here. That's the mummy. Becomes infected and resists microbial decay, so it can hang around in the soil. When conditions are right, they spring up and produce these structures that produce spores that then can propagate the fungus and reinfect the caterpillars. This is a big, big business collected in Tibet. You may have seen these as enhancers of various kinds. They're tonics and are available in the market. And in fact, this one was purchased in Chinatown fairly recently. One of the curious things that I think is perhaps important, these are bound up with red string. These, I don't know what color. This is from an old Chinese herbal type book. But this red is good luck. It's an auspicious color. And in our collection, I can go back to materials that were purchased in China in the 19th century, and they're bound up in red thread. So it's a common theme here. So these are referred to as mummies, these parts. They're mummified, and that has to do with the way in which the fungus grows, the compounds that are produced, and the fact that they are resist microbial attack. And some of these are quite spectacular. In this group, a tarantula that has its fungus disease. It's long gone. It's long gone now. Now, if you go upstairs into the glass flowers, there's a case now that has some of the decayed fruit models by the Blashkas. And those are also showing you mummies. And here's a mummified fruit. These are stone fruits, peaches, in this case. And these mummies, I give you a life cycle here. No exam. You don't have to know this in the end. But I want you to think about this in terms of the biology of these particular mummies. The fruits become infected over here. Those infected fruits have the fungus growing throughout them. They are structures that are set up to overwinter, so they can fall to the ground. In this overwintering, they need to be able to resist microbial attack. That is, they can't just be overgrown by other fungi. So they have some chemical defenses. They make it through the winter over here. They produce fruiting structures that begin this disease cycle again. So here are mummies, mummies of a different type. And look upstairs, you'll be able to see those. I want to revert again to thinking about a lichen because I have to get in this other M, Manna. And there they are, gathering manna. And, you know, the Israelites moving through, trying to get out of Egypt, and they're provided with food. And this food appeared kind of mysteriously in the mornings, especially when there was a heavy dew and so forth. And here's collecting manna. And what does that have to do with the lichen? Well, there happens to be a lichen that's called the manna lichen. Here's some of it. These are quarter-sized pieces of the lichen. These, when they're dry, they look like pebbles. When they're hydrated, they get soft and squishy. And some people would argue that this name, manna, like coriander seed, white in its taste, was like wafers of honey. And Moses said, keep some of this. Show future generations what's happened. And I think this is really the first injunction to keep a herbarium. Okay, so keep some of it so you can prove what you've got. They're voucher specimens, we'd call them. Okay, so manna. Maybe the lichen is manna, maybe not. But certainly it was named in such a way that might make us think so. Aspergillia esculenta. Esculenta. Good to eat. All right, so I'm kind of going through diversity here. We open a cabinet and what's inside? What's inside is a different kingdom. It's a different group. And we'll go all the way over here to these bacteria. We have collections of bacteria, and they were cryptogams at one point. They were referred to as cryptogams. And here's one of these that is really tied to the Farlow and the Farlow collection. It's a member of the Myxobacteria, and these are really special organisms. Bacteria are small, small cells, and these cells glide and come together and mound up and form something that looks like this. Looks like a fungus in a way. But it's not. It's made up of these aggregated bacteria. And these were studied by one of our botanists, Roland Thaxter. And I've talked about archives and materials and so forth that we are able to use and to have because of these collections. But what I'm going to show you next is a film clip of Roland Thaxter himself, taken, it's 1920 something or other. And it has to do with where we found this. We found it in the film roll in a storage room in the basement, and now it's digitized, and now it's here for us to see. See somebody who I never thought I'd see, died in 1932. Whoops. Here he comes, out in front of the Farlow building with his dog Bobby. And Bobby seems to be pretty well trained. And they're up to the front of the building, C.W. Dodge, Curator. Aha, something good. And he comes inside, and I always like to say, he almost smiles. Now, he's very special. Not very many of us could say that we've put several groups of organisms on the map, but he did. He discovered these. He defended them in the sense that people thought they were something other than bacteria. He illustrated them. He did the first work on the diversity of these. But, you know, this wasn't the only thing he did. He described about 1,000 species of insect parasites in the Labellobeniales. He did the first monograph of the insect parasites in the Entomophthalales. He also worked on the Endogenoceae and produced the volume that we used up until the time I was a kid. So here's some of his illustrations, and he's an excellent draftsman, an excellent artist. And he's illustrating all of these myxobacteria that he went out into the field and saw and described. There's a myxobacteria group these days. They get together and have symposia every couple years. He's their patron saint of that group. And then there are other bacteria that we have. And the cyanobacteria, many of you may know these as the blue-green algae, but they are part of our collections. And here is an example of them. This little smear here is one of the collections that we have, and one can take some material from here, make a microscope mount, or you can use this mount that they've already made here. And this is really curious. It's an old specimen. And the way that the mounts were made were with mica chips. So a piece of mica with the specimen mounted on it. And then it could be used under a microscope. And this is a drawing from the herbarium of one of these. And then I happened to notice this sign on a website for the Charles River Association warning people to stay away from water that is scummy and green like this because it's filled with these cyanobacteria that are toxic in various ways. So if we are looking at documenting, thinking about these organisms over time, we can go back and check many things by having these records in the herbarium. And I'll talk about that a bit more later as well. Well, we'll bounce back over here. These are the eukaryotes and the Stromena fills. These are organisms that, when I was a student, we thought were fungi. We thought they were fungi because they have hyphae, they have spores, they have various characteristics that make them look like and behave like fungi, but they're not. They belong to an entirely different group, along with some algal groups that are over here. And I wanted to bring this up because there's probably no fungus-like organism that's quite as important as this, Phytophthora infestans. Phytophthora infestans causes the late blight of potatoes. And if we want to track back some of the major immigrations, major movements of people, famine and death, we come back to this always as one of the examples. In Ireland in the 1840s, outbreaks of this potato disease began to happen. They became more and more severe over time. And at that point, the idea that an organism caused the disease was not well established. This idea of an organism causing a disease of plants seems so intuitive to us, but it wasn't. And there were great debates. Why were the potatoes dying? And the potatoes were dying at such an extent that people didn't have food, but maybe it was the environmental conditions. It rained too much. Maybe it was genetic, that we'd been growing these potatoes so long they just gave up. Many explanations here, but in fact, it was this fungus-like organism, Phytophthora infestans. And I bring this out because it's one of our collections that has been used in modern times to help explain the epidemic and what the strains were and what the genetics of those early Phytophthorus were out in the world. And this is from the Thomas Taylor Herbarium. Thomas Taylor was an Irishman. He was trained as a physician, but he loved these organisms. He loved mosses and hepatics and fungi, and he studied them. And as the famine began to take over, he took up his profession as a physician again. He died in 1848 of the famine fever, so he didn't last long. But here's a collection from that herbarium. Here's a little piece of the leaf that researchers are able to take a portion of it, do sequencing of a particular region that has to do with its mating type, assign that to a group within the organisms, and be able to determine what the race or what particular population of Phytophthora was there at that point. So we can tell what that was like in Ireland from these studies. Here's the drawing by Thomas Taylor showing the organism on the leaf surface, little description here, various other documentations. So here in Cambridge, we have a memorial to the potato famine. And this, never again should a people starve in a world of plenty. This disease is still common. There's still big outbreaks of it. We're still trying to understand the genetics and the population structure here. And this is from a recent paper. Our colleague who's worked with us in the herbarium, Jean Restaino, has sampled our collections, and they've put together from all of their collections a kind of map that shows us where these various strains are present, and begins to think about where it is that these came from and how they were introduced and what the genetic changes are over time. So here it is. Collections that we have today can document epidemics of the past and inform us about the future, about how we can think about these populations. So finally, something green, mosses, and I love this piece this statement by Ed Wilson. So, this symphony, this group, this looking at these organisms, understanding them. And the example here of just thinking about collections and what they mean and how pertinent they can be is one that our curatorial assistant, Genevieve Lewis Gentry, turned up a while ago. And this is a collection made by Thomas Potts James. He ended up living in Cambridge here. But at this point, he was a volunteer with the Union Army, and he was stationed around the White House. And so here it is, 1863. He's there. And isn't it curious? He's collecting mosses, you know. How dedicated is that? You're there collecting and working on these particular mosses. And because the Civil War is so well documented these days, we can tell what Lincoln is doing and who he's meeting with. But because our collections are so well documented, we can tell what Thomas Potts James was doing that day. He was out there looking at mosses. Whatever he was writing home about, he was looking at mosses. So for me, one of the most interesting things with these collections is to look at them and realize that these are human endeavors. Somebody went to the far ends of the earth. You'll see some examples from Antarctica upstairs. Somebody went there, and while they were there, they collected organisms, and they brought them back. And they documented the part of the surface of the earth. And when we think about that, it's really quite phenomenal that we have these records, we have these documents, we have things that we can say about the people and the organisms, both. All right, a little more green, it's okay. I want to mention the algae. We have a big collection of algae. And along with a number of other parts of the collections where we're working on fungi and lichens and bryophytes and algae, we're doing a digital project. And this is a screenshot from the macroalgal portal. And what this allows us to do is to look at collections from around the world that have been digitized. And we get collection data as well as, in this case, beautiful examples of the algae themselves. And these are seaweeds. And you can construct a map. And here's a map with a few of these here. And if you can tell, here's Long Island Sound, Cape Cod. And you get a distribution pattern here that's very clear. Most of those dots are south of Cape Cod, with only incidental ones above. And this is a pattern that we see with the distribution of marine algae. That Cape Cod is a dividing line. Southern things are below, Arctic things are above. And it has to do with the Gulf Stream and the water temperature across the Atlantic. So to go back to Farlow and to think about this a little bit, my former assistant Ingrid will love this one, Anasquam. This is from Farlow's Marine Algae of New England, 1879. And he has this little bit here about the Goose Cove in Anasquam. And the fact that he could collect there, he could go and look, and what he found was algae that should be south, Cape Cod. And he began to think about this entirely different flora in this one spot. And if we kind of extrapolate now, we can think, well, this is something to do with climate change, maybe, with environmental change. And so with these algal specimens that we begin to see, we can plot out distributions. And because we know that there are these dividing lines, we know the temperature is really an important factor here, that we may be able to use these, and people are using these, to look at and evaluate what it is that has, what climate change means. But it's an interesting little piece of observation that he makes. The Arctic versus the Southern, and these sharp lines that he was able to note. But a great observation here, a great observation about the organisms. All right, well, you may be getting tired, but I want to show a couple more examples here because the diversity is so wonderful. And these are the myxomycetes, or the mycetic zoa. If you open a textbook on mycology, you will always find that these are treated in that book. And that's because by tradition, they're studied by mycologists. But by phylogeny, as you saw, they're in their own little group. And many of you have seen these. They sometimes are big, they sometimes are tiny. This is a sketch from Roland Thaxter's sketchbook, and this is kind of supposed to be natural size, so little tiny things with a sporangium and spores here. And one of the curious things about these is that the spores germinate, and they produce these amoeboid type cells. They're swimming, some of them have flagella, some of them move like amoebae. And they aggregate, they get together, and they form this clump of plasmodium. It's not cellular, it's just a big lump of cytoplasm with lots of nuclei, and it's capable of unidirectional growth. So from here, out, growing out, ever expanding outward. And you see these sometimes in the woods. You can find them on the forest floor. They're very bad fungi, if you wanted to put them there, because they don't have hyphae. They don't have any of the structures other than spores that fungi have. And there are a number of different groups of these amoeboid-type organisms that we have represented within the collection. And this is another of Haeckel's drawings of these. They're just fantastic organisms to look at. So, what do we have here? You thought everything was okay, but you'll get over it. You'll be confused soon. We have all these different types of organisms in one collection that's under this one roof. It's not like the MCZ, those are just all animals. It's not like the other herbarium, those are all plants. No big deal. Here it is, multiple kingdoms in one unit. And these are widely diverse organisms. They're diverse in their phylogenies. I've used the phylogenies to kind of pinpoint where we are. But they're also highly diverse in terms of their biology, what they do in the world. And we know that they're important in many ways. Ecosystem function, that is how materials are cycled and recycled. In human health, I've pointed out a couple examples here. Food security, are the potatoes safe? And I think the overall theme is that they're mostly understudied, that there aren't a lot of people studying these various groups. And I pointed out with fungi, for example, that some of the estimates are that there's 1.5 million or 2 million species of fungi that exist in the world. Some estimates are even higher. But we've only described about 75,000 to 80,000 species. So it's a long way to go. So all of these are important organisms for us to be able to know about, to be able to study the diversity, to be able to understand what they're doing out in the ecosystem world. So I've emphasized a lot of history here, but I wanted to point out that there's a whole lot that is going on in the building right now, and I'll just mention some of those. Diversity and Life History Studies of Ladlo Benielis, one of our graduate students, lichen systematics, Michaela Schmuel, orbiliomycetes and ultrastructure, Roseanne Healy, and I are working on those. Fungi of Boston Harbor Island, spearheaded by Danny. Documentation of early collections from Venezuela, Teresita Turiaga. Systematics and life history of ascomycetes. That's the only way I figured out I could talk about my own stuff. Inventory and comparison of truffles and mycorrhizae at Arnold Arboretum and Harvard Forest. That's Roseanne Healy again. And projects to digitize the collections, and I mentioned those. And we have a whole batch of the digitizers and the people working on these right out in front here. And this part, I want to make sure that you've picked up. There's a yellow sheet on the table on the side that talks about our workshop that we're holding on the weekend. That workshop on Saturday is about how to use these digital collections, how you can help us use and organize these digital collections. So we are digitizing, capturing certain kind of minimal data, and then those go out onto the web. And then, if you really want to get involved here, you can look at those and help transcribe labels. And so that's what this crowdsourcing, that's what we're going to be talking about on Saturday. And we welcome you to come and help us with that. And that is largely sponsored by our friends group, the Friends of the Farlow. It's 34 years old, has supported the library and herbarium over those 34 years. There's over 30 annual lecturers that we've brought in as the Friends of the Farlow. 20 or more graduate students have come to work in the collections and improve their dissertations and so forth. About 10 visiting scholars have been here on the Harvey Pofscher Fund, which sponsors faculty members. And then a volunteer group here who have helped to set us up and keep things going tonight. Thank you. Now, not yet, not yet. I want to reflect a little bit. Linnaeus, our forefather in a way of thinking about collections and thinking about names and naming, in his Philosophia Botanica, talks about travel. And I think we can kind of metaphorically think about this in a larger sense of what it means to collect, describe, and work on these collections. The starting point must be to marvel at all things, even the most commonplace. The means is to commit to writing things that have been observed and are useful. The end must be to depict nature more accurately than anyone else. So these are kind of marching orders for us as we think about these groups that are highly, highly understudied. And then I want to thank the curatorial staff, and I've mentioned several of the people who have been involved here. Lisa de Cesare is here. She kind of helped us spearhead part of the exhibits, but Jen and Michaela found a lot of the specimens that you'll see upstairs, and all of the people who have worked there and been in the collection for all these years. And I'll leave you with a Roland Thaxter illustration and a little bit of Henry David Thoreau. Thank you.